It provides attachment for RNA polymerase. Regulator gene, that is I gene, it synthesizes regulatory protein regularly. And next, operator gene, it receives the product of the regulator gene. The regulator gene will produce a protein which is a regulatory protein. It consistently produces a protein. This protein will be received by the operator gene. Here if you look at very clearly, this is the operator gene which receives this particular protein. So let me talk about structural gene. Structural gene is the gene which is converted to mRNA, that means transcripted to mRNA, not converted, better you say transcripted to mRNA, means the information is copied to mRNA, and further mRNA is used for polypeptide synthesis. So that is called structural gene. And structural genes are very important. They regulate the metabolism. Now in a lac operon, you can see one promoter gene, one regulator gene, one operator gene, and three structural genes, Z, Y, A. When a bacteria is present in an environment where lactose is absent, this kind of operon structure we could able to see, where the repressor protein which is produced here will go and bind onto the operator gene, and they will not allow the RNA polymerase enzyme to transcribe Z gene, to transcribe Y gene, to transcribe A gene. So this operon is switched off. It is switched off in the absence of lactose. So the operon is switched off when the repressor protein or regulatory protein binds with the operator. This kind of regulation is called as negative regulation. When it is doing negative regulation, the regulatory protein is called as repressor protein. Right? Then we'll start discussing about structural genes. I am just revising once again for your information. Z gene, there are three structural genes you can see here, Z, Y, A. Z gene is a structural gene which codes for an enzyme beta galactosidase. The beta galactosidase will break down the lactose and convert it into glucose and galactose. And Y gene is a structural gene which produces an enzyme permease. The enzyme permease will allow lactose into the cell. A gene is a structural gene which produces an enzyme transacetylase. Transacetylase adds acetyl group to the lactose. Till now, we do not have the significance of the acetyl group onto the lactose, addition of acetyl group onto the lactose. It is meant to protect the lactose to be digested by other enzymes. That is only the significance. Let's talk about when a bacteria is present in a media containing lactose. Lactose enters the cell, and the lactose which enters into the cell bind with the repressor protein. Actually, this regulatory protein or repressor protein is meant to bind with operator. But when lactose is present inside the bacteria, the lactose binds with the regulatory protein or repressor protein and do not allow the repressor protein onto the operator. Then the operator becomes free. When the operator gene becomes free, the RNA polymerase can access the promoter sites and start the process of transcription. When it initiates the process of transcription, it transcribes Z gene and produces beta galactosidase, transcribes Y gene, produces permease, transcribes A gene, produces transacetylase. In that way, the operon becomes switched on and the lactose metabolism will run inside the cell, digesting the lactose into glucose and galactose. And as soon as this glucose is formed inside the cell, the glucose is utilized for energy production. This is what. So then this particular situation, we call this as positive regulation. Positive regulation occurs when the operon is switched on. That too when lactose binds on the repressor. Here, to make the operon switch on, the only inducer is lactose. This operon is a inducible system because it is induced by the presence of the lactose. That's why we call inducer is lactose. So don't mistake, I means inducer. I is derived from inhibitor term. Always inside the cell, there is a very low level of expression of lac operon will be there. Otherwise, lactose will not enter the cell. This is the positive regulation and negative regulation. So 2-3 important things we have studied in the lac operon, that I gene continuously produces the repressor all the time. That's the reason the genes which continuously express all the time are called as constitutive gene. In the lac operon, there is only one constitutive gene, that is I gene. In the lac operon, there are structural genes which are non-constitutive. They will express only in the presence of inducer, that is lactose. They will not express when the lactose is absent. That's why ZYA gene are called as non-constitutive genes. In the lac operon, not only lactose works as an inducer, but other lactose members also can act as an inducer, can bind with the repressor protein and can help in the proper transcription of ZYA genes. One more important point to be noted at the end, that you take any operon, that operon is going to be very specific in terms. One second. Sir, good evening, sir.
Good evening. Sir, auditor session.
Extremely sorry. Okay. Now this is what each operon has specific operator. I said each operon means lac operon has a specific operator. Lac operon operator cannot receive the repressor protein of val operon. Val operon operator cannot receive the repressor of arabinose ara operon. Point to be noted. These are very important points. Like each operon will have specific operator and specific repressor. That means the lac operator which is present only in lac operon and specifically interacts with only lac repressor. So this lac operator cannot interact with the repressor of other operon. So this operator and repressor are very specific for each and every operon. This is the point which is to be noted. You may ask, sir, whether the promoter is common. You may also ask whether the structural gene is common. Absolutely not. Yeah, the structural gene is also specific actually. The ZYA structural genes are there, 3 genes are there in the lac operon. In other operons, there are different structural genes. Like tryptophan operon, there are 5 structural genes. Similarly, in other operons, the structural gene number may be different because prokaryotes are polycistronic in nature. Right, students? Yeah. Coming to the question which I have given to you. That question will give you an idea, very clear-cut idea, that which enzyme will be produced in a cell in which there is nonsense mutation in the LAC-Y gene. This is based on the knowledge of lac operon enzymes produced by the structural genes, and also it emphasizes on the mutations. That too, nonsense mutations. Nonsense mutations are the mutations which results in stop codon, UAA, UAG, UGA. When Y gene, in Y gene, nonsense mutation occurs. I have explained you in the previous class. So here, if you look at up to Z gene, the transcription occur. In the Y gene, the mutation results in UAA or UAG or UGA, which ultimately stops the process of transcription. So you will get only beta galactosidase enzyme. So according to this explanation, the beta galactosidase enzyme is the correct answer, right? This is what we have discussed in the previous class. I think so. I am clear with the lactoferrin. Additional information, as usual, we are going with regular information of the NCERT, then package, and then additional information later after completing the chapter. So after this, we will take up HGP. Are you ready? Hello. What happened? Again,
Yes, sir.
Yes, sir. I
Yes, sir.
think people have exam, huh? KNMS, all the KNMS students. Okay, anyway, I'll carry on. I change it to something. So we'll take up a new concept, HGP, Human Genome Project. I don't know how many are very interested in Genome Project. Anybody know about this? Do you have any preliminary or previous idea of HGP, Human Genome? Anybody from the class? Right. Anyway, no problem. We'll take care now. So before jumping into that concept, I have few questions. Again, these questions to help me where you are standing in the concept, and it also helps you to understand the concept little thoroughly. Okay. So you just freely interact. I just want what is meant by genome again. I just need a particular answer for genome. What is meant by genome? Let's discuss little in detail manner what is genome. So before entering into the Human Genome Project, it is it's very necessary to you people have a good idea of what is genome. Am I right or no? So please, can you help me? Anybody? What do you feel? What is meant by genome? Once again, I just wanted to help you out. What is meant by genome? Then, if you know the genome perfectly, then you can understand what is Human Genome Project. Anybody from the class? It's open.
Genes.
Genes. Okay, then we should not give the answer like a layman in biology. Already we have been taking this genome concept now from 11th class. You just tell me the technical part. Now you are doctors, feel like a doctor now. One more year to enter into the medical college. Just 2022 you are into the medical college. So genome means, let me know what is actually genome. So genes, okay, and okay, you tell me about the genes. Which genes? How many genes make up a genome? If you interact, then it will.
Haploid number of genes.
Haploid number of genes. What is haploid? Why not diploid? Okay, see, I will just define from my side. Single set of chromosomes. Always when we try to define a genome, we have to define in this manner. Single set of chromosomes. This is haploid. Then single set of chromosomes is one genome. Then we say haploid, N. Two sets of chromosomes, two genomes, 2N. Three sets of chromosomes, three genomes, 3N. Four sets of chromosomes, four genomes, 4N. Five sets of chromosomes, five genomes, 5N. So, but he said Human Genome Project. So he is working on a particular genome. Which genome? Whose genome? Human genome. Now, can you tell me human genome has how many number of chromosomes, or how many, instead of chromosomes, I need base pairs. Can you tell me number of base pairs in the human genome? Sir, I am in a class. Can I call you later, please? Sir, I will call you at 8:45, sir. Thank you. Please tell me. Number of base pairs, Geetika. Ajitesh, Amrita, Ananya, Sarvesh, Nandini. Help me please.
3.3 into 10 power 9.
3.3 into 10 to the power of 9 base pairs. These are present in how many genomes?
N, sir.
1 genome.
One genome. Then
Genome.
2n is equal to
6.6.
okay. My question. Okay, I will not ask here. Let me move ahead. Okay, this is what genome. See what actually the Human Genome Project is. Right. Now, let's understand the goal of the Human Genome Project, the main goal. So the main goal of the Human Genome Project is complete mapping and understanding of the genes of the human being. So I'll start with the goal. The goal is very simple. Complete mapping. And understanding of the genes of human being. This is the main goal of the Human Genome Project. Clear? Now this project, what is here before going into the project, what is mapping? Mapping is locating the genes on which chromosome they are. You have studied that humans consist of 2n is equal to 45 or 46? 46 chromosomes. That means n is equal to 20, 2n is equal to, I can say, 23 pairs of chromosomes. I said 23 pairs. Means, for example, a gene causing Alzheimer's, a gene causing cardiovascular disorders, a gene responsible, for example, a gene responsible for any other diseases, or a gene responsible for a particular character, where they are located, on which chromosome? First chromosome, second chromosome, third chromosome. In genetics, we have studied about mapping of the genes on the chromosome. Mapping of the genes on the chromosome, which is given by Alfred Sturtevant, that is one of the important innovation discovery which helped in Human Genome Project to map the genes. So the first thing, complete mapping and complete understanding of the genes which are present in the human being. Now this Human Genome Project is a mega project. Why we are calling it as mega project? Because it is a 13-year project. And it is launched in the year 1990. You know very well, the two individual human beings, do they show any difference in their nucleotide sequence? Any idea about that? Do they show any nucleotide sequence difference? So we say that 99.9% of the individual's DNA is almost all similar. There is only, I think, 0.1 difference between two individuals, right? And the difference of two individuals will be reflected in that 0.1 only. Difference in the nucleotide sequence. Now, taking this particular difference into consideration, this 0.1 difference, what do we understand? Actually, 0.1 difference in what way it is going to be helpful? In 0.1 difference is going to be helpful in identifying the characteristics of one individual and comparing the characteristics of one individual with another individual. So the two human individuals are different. They are showing variations because of difference in 0.1% of nucleotide sequence, difference of 0.1%. So in order to understand that particular thing little more clearly at the molecular base level, this particular human genome project is launched. The ultimate goal of the human genome project is just to know the genes and understand the functions of the genes. As the human genome consists of 3.3 into 10 to the power of 9 base pairs, right? This is going to be a mega project because you have to sequence out 3.3 into 10 to the power of 9 base pairs. You have to sequence, you need to know the sequence. And this mega project is coordinated by US Department of Energy and NIH, which is called as National Institute of Health. So this is coordinated. US Department of Energy. This Department of Energy is called as DOE. And National Institute of Health, which is called as NIH. Later, Wellcome Trust became a major partner in the Human Genome Project, and later Japan, Germany, France, China, they became major partners in the Human Genome Project. Then this project is completed in the year 2003. So this concept is completely knowledge-based concept. And there are some places are there where you have to understand the concepts. Like I will help you out. It's more information. It is information based, knowledge based. Then why we are calling it as a mega project? There are certain reasons to call it as a mega project. So HGP called as mega project due to the reason one here, we have to sequence almost all 3.3 into 10 to the power of 9 base pairs. To sequence one base pair, to just sequence one base pair, and to know the sequence of one base pair, you know how much dollars have to be spent? $3. Proportionately, the total estimated cost to sequence the total base pairs present here is going to be $9 billion. This is the estimated cost actually. That is the reason this is called as mega project. And you cannot ignore this. Why? Because the questions are based on this. The Human Genome Project is called as mega project because of the following reasons. So these are profound, distinct reasons. That's the reason this HGP called as mega project. Second, I already told you the cost. And I'll come to the number of base pairs that we have to identify. Just now I told you 3.3 into 10 to the power of 9 base pairs. That is very large number of base pairs to be identified and sequenced. Second. This is one more reason to call it as a mega project. Here it requires large number of scientists, technicians and supporting staff. This is also the reason. Then here the storage of data, that means whatever the data is going to be generated by sequencing this, it really requires 3,300 books. With each book comprising of 1,000 pages, with each page minimum comprising of 1,000 typed letters. So this is going to be very huge. That's the reason it is called as mega project. Storage of data. So all this knowledge base, we have to take it little more sensitively. We cannot ignore each and every point, right? Because it's about entrance exam. So we cannot even, ignore the points here. So because each and every point become very important, because these are all the reasons for calling this as a mega project. So 3,300 books to, store the data. And, they estimated that each book must and should comprise of 1,000 pages. And each page 1,000 typed letters. That's the reason this is called as mega project. Then luckily, the point here, one important point we have to make a note here. So this is replaced by high-speed computational devices. So this problem is resolved by using high-speed computational devices. Where they help in managing the data. And moreover, apart from these reasons, because of these things, we are calling it as the mega project. One more important thing is that during this Human Genome Project, a new science have been developed. That is called as bioinformatics. So during HGP, bioinformatics science developed. So these are the reasons that we call this HGP as a mega project. Then what is bioinformatics? You might have doubts. So bioinformatics deals with the information of science, managing the data of the science, that is human genome. Not only Human Genome Project, it manages the data of chemistry, it manages the data of mathematics, it can manage the data of molecular biology, and it can retrieve the data, it can organize the data, it can analyze the data by using computational methods. So bioinformatics is a science which is developed during this HGP, which is useful, in managing, organizing, analyzing, retrieving the data related to the various areas of the science. So this is given as a course after you complete your bachelor degree. Then this can be chosen as a separate course, which is called as bioinformatics, which is in integration with the computational methods and computational techniques, right? So this is about mega project. So I'm done. So once again, I just wanted to revise. Sequencing of complete genome requires $9 billion, where cost of 1 BP is $3. Large number of base pairs to be identified. Large number of scientists and technicians supporting staff are required. Storage of data required 3,300 books with 1,000 page, each page 1,000 typed letters, but that can be compensated by high-speed computational devices, which helps in managing the data. And during this course, the bioinformatics science has been developed. I told you what is bioinformatics. Now let's move to the goals of HGP. Anyway, why it is called as mega project, and it is a 13-year project, as I told you, 1990 to 2003. And who are included, that means who carried out this project also we have discussed, US Department of Energy and what is NIH? Okay, then there are certain other countries also gave a hand in the Human Genome Project. Then we'll come to the goals. The main goal is minimum identification of approximately 20,000 to 25,000 genes in human DNA. This is the first goal. $20,000 to 25,000 genes in human DNA. Second, here the main goal is to sequence 3 billion base pairs of human DNA. Determine and sequence. Third, to store the information in database. Fourth, to improve the tools for data analysis. See, in order to analyze the data, so the tools have to be improved. This is one of the important goals. These two points are almost interlinked. Then whatever the data we got, that data should be transferred to some other sectors such as industries where this data which we got by the 13-year mega project is useful in providing helping out producing products for the human welfare. So it is about transfer, which we call it as technically transfer related technologies. So this data which we have produced during this 13 years, it should be transferred to other sectors such as industries. To solve ethical, legal and social issues. This is one of the important goals. So ELSI, this is called as, what is this ELSI? See, Human Genome Project. Na? Which human genome they have collected is the question right now. Whether they collected scientist human genome, whether they have collected any other person's human genome, whose human genome is collected. Here samples are taken from wild, populations, groups. It's not a single person. And from wherever they have taken the samples, they followed. There are some, to do an experiment or to perform a project related to this kind of, involving humans or involving animals. There are so many, things we have to take care. We have to bound to the rules and regulations, norms. Ethical, there are ethical norms are there, legal norms are there, social, ethical, legal, social issues have also to be considered. You cannot, give out for example, there is a population residing in a particular area, there is another population residing in a particular area. You take out that particular genome and you analyze it and you got the result. They are showing this kind of particular, means specific characteristic features. and you have got that population result also. They are showing that some particular sequences, some particular characteristic features. Here we cannot pronounce it, or we cannot announce it outside that these populations are inferior and these populations are superior. So there are so many ethical issues, legal issues, social issues. We cannot easily collect the samples, and we cannot do the project that easily. So this here the main goal of HGP also is that they also have to take care of the ethical issues that are going to come out during this project or after the project. Legal issues, social issues, they have to take care, which is called as ELSI, LC, right? To solve ELSI, ethical, legal, and social issues. So to carry on an experiment, particularly now, anyway, I think you have been you have heard about these kind of what trials, clinical trials. So clinical trials, you need to take permission from the individual on whom you are going to try. So clinical trials have so many rules and regulations, right? So all these are related to the ethical, legal, social issues. Here also, one of the important goal of HGP, they have to take care properly the ethical issues of this project, legal issues of the project, and social issues of the project. Then the next goal is the sequencing. They sequence it not only the human, but initially they sequenced non-human model organisms. They sequenced a nematode pathogen. They sequenced the DNA of the nematode pathogen. They sequenced Arabidopsis. They sequenced rice. They sequenced yeast DNA. They sequenced Drosophila. They sequenced bacteria. Like non-human model organisms, they sequenced it. That is one of the goal of HGP. Sequencing of non-human model organisms. Understand this. What are those non-human model organisms? Let's start. Let's start from the prokaryote. Bacteria. Right. So let me write down after their sequencing, how many, that is how many genes they have sequenced, how many genes are present. Instead of genes, let's talk about base pairs, how many base pairs they have sequenced in each and every model organism, non-human model organism. Let's start with bacteria. I'll take E. coli. So this is not necessary that you have to remember this. This is just an idea. They are giving you an idea that in E. coli, I think they have sequenced 4.7 million base pairs and they found out 4,000 genes. 4.7 million base pairs. You have already written the count of the E. coli base pairs. 4.6 into 10 to the power of 6 base pairs. Am I right? Hello, are you there in the class, students?
Yes, sir.
Right. Then they sequenced yeast, especially Baker's yeast, Saccharomyces cerevisiae. This is Escherichia coli. This is Saccharomyces cerevisiae. In Saccharomyces cerevisiae, they found almost all 12 million base pairs. In that, they sequenced out, that means they found out 600 genes. Third, Drosophila melanogaster. This is the fruit fly. This is an experimental organism which is chosen by Thomas Hunt Morgan, father of experimental genetics. There here in the human genome product, they sequenced almost 180 million base pairs. They know the sequence of Drosophila melanogaster, complete sequence. And they found out 13,000 genes which are actively working. Genes means these are expressive sequences. They are expressing and they are performing a particular function. You have to understand this. Then a nematode, Caenorhabditis elegans. In the case of Caenorhabditis elegans, 97 million base pairs. And 18,000 genes they have discovered. And there are some other non-human model organisms are there. This is little extra information out of the NCERT, which is approximately it will be majorly you have to go remember for Drosophila melanogaster 180 million base pairs of 13,000 genes. And there are two more like Oryza sativa, 430 million base pairs where they found out 32,000 to 50,000 genes. Then Arabidopsis 130 million base pairs with 2,500 genes. So at this level, I can give you only the data. And anyway, this is also provided in your package, right? You can check it out. Where 2,500 genes they found. And in Oryza sativa, which is rice, they did 430 million base pair sequence. In the data set, it is 32,000 genes to 50,000 genes. So these are the non-human model organisms. These are the goals of HGP. All right. So once again, I just wanted to revise the goals. So because I told you some of the reasons for mega project. What is the first goal? Anybody? Let's revise. Please help me. Abhirami, what is the first goal? Achyut, what is the first goal?
For identification of approximately 20,000 to 25,000 gene in human DNA.
Very good. Sarvesh, what is the second goal? Okay. Denilda, what is the second goal?
Sir, to determine and sequence 3 billion base pairs of human DNA.
Okay. To determine, right. What is the third goal, Aishwarya? Very good.
To store information in databases.
Okay, Pranav, it is 25,000. Now, have you checked it properly? Is it 25,000? Okay, I'll correct.
In the second one, it is 6,000, sir.
Not 600. Okay. Right, corrected. Then what is the third goal? Nandini?
To store the information in data.
So the fourth goal, Jayanirudh? Okay. What is the fourth goal, Amrita?
To improve the tools for data analysis, sir.
Next goal, please tell me. Fifth goal also you tell me, Amrita. What is the fifth goal?
Transfer to other sectors, such as industries.
Also, sixth goal. Can you tell me what is the sixth goal?
To solve ethical, legal, and social issues.
Sylvia, what is the seventh goal final?
Sequencing of non-human model organisms.
Ajitesh, the Drosophila melanogaster. How many genes are identified?
The 13,000, sir.
Yeah. So these are the goals. So we are done with the first part of the Human Genome Project. Then we will discuss the process. Process means the methodology. How we are going to sequence. How they scientists sequence the human genome. So which we call it as methodologies. We'll start up with methodologies. There are two approaches, right, to sequence the human genome. The first approach is called as expressed sequence tags. Let's first start discussion on expressed sequence tags. So this is really a wonderful approach, right? I'll tell you. So what did they actually do in this particular methodology? For example, there is a cell in which this is an E. coli. Now all the information present in the DNA which is going to be expressed will be expressed as RNA first of all. Why? Because it will be converted to RNA. You will get mRNA. So if you take mRNA, if you take only mRNA, identify the genes which are expressed as RNA, then that is sufficient. You will get the total number of genes present in that particular organism. This is what they did in the first methodology. They simply identified the genes which are expressed as mRNA. So that's why this methodology is called as expressed sequence tags. And point to be noted here again, we are not discussing the process in the expressed sequence tag. They simply isolated the mRNAs from a cell and finally they sequenced the mRNAs, right? Then if they sequenced the mRNAs, we'll get to know the DNA sequence now. Finally, they found out the number of genes present in that particular organism. So this is what called as expressed sequence tags. Any gene which is going to express, that will first convert to mRNA. Then mRNA will be translated to synthesize a polypeptide chain. So here, identifying all the genes that are expressed as RNA is called as express sequence tags. Identifying all the genes that are expressed as RNA. This is one methodology they applied. Second methodology is called as sequence annotation. So just follow the steps which I am going to give you here, right? Sequence annotation. So in the case of sequence annotation, this is about sequencing of the whole genome. Sequence annotation means this is sequencing of whole genome. Not only express the sequence tags, here the whole genome is taken into consideration and it is annotated. Annotated means identifying the sequence and assigning functions. Okay. One second. For example, you found out a sequence of a gene in chromosome number 1. You have to assign the function. You need to find out the function also. Then, in order to sequence, what you have to do? Let's start. The first step for sequencing. See, first you have to isolate the DNA. See, in the case of human genome, what they did, they collected blood samples. Mostly what they did, they collected sperms. Sperm contained a haploid genome, complete haploid genome with X and Y. Why? Because why not the other tissue cells? The only reason, in the sperm, there is more content of the DNA when you compare with the protein, and isolation of the DNA from the rest of the protein will become very easy because the protein content will be less in the sperm cell when you compare with DNA content. When you take other cells now to isolate the DNA, there the protein content will also be very high. To isolate the DNA, it is going to be a very tough task, not that easy. Now you have to imagine at the molecular level. It's not that the content level. Content will be given, then we'll by heart the content. That's a different issue. But here you need to focus on the techniques that are involved in the isolation. First of all, to sequence, you need to have the DNA in your hand. The DNA you cannot take it in the hand. Cell is micro, completely very, very small. From the cell, you need to isolate the DNA, take out the DNA only. Yeah, we don't want any. How do you know that in a cell, if you take a particular cell, it's all fluid, na? In that you have to identify which is DNA, which is protein, which is fat, which is lipid. So biology is more over techniques. Isolation techniques are very, very important in biological field. Now here, first of all, you need to isolate the DNA. Telling is very important and remembering is also, telling is very easy and remembering is also very easy. Isolate the DNA, okay, that's it. Isolate DNA is not that easy. That in a cell, DNA is there, proteins are there, so many things are there. Then point also to be noted logically, then which cell they have taken. They have collected so many human samples and those names are not leaked out, because I told you in the ELS side, ethical issues, legal issues, social issues are there, which are answered properly in the Human Genome Project. That is also one of the goals. And they have collected sperms specially. They have isolated the DNA from the sperms rather than males. For males, they have collected the sperms. And then from females, they have collected some white cells. So let's talk about males. That's easy, where the sperm has more amount of DNA. It will contain one genome, 3.3 into 10 to the power of 9 base pairs. Then you have to lyse the cell. The cell has plasma membrane. By using lipases, you need to lyse the cell. After lysing it, whole cytoplasm along with protein, nucleic acid, everything comes out. In that, you have to isolate the DNA. If you take sperm, the DNA content is more when you compare with protein. So isolate the total DNA from the cell. First of all, first step to sequence, isolate the total DNA from the cell. Now I am also giving you a hint from which cell also I gave you. Most of the times this project is isolated the DNA from the sperm cells of the male. In sperm cells, there is a thing that in female, 2 X chromosomes are there. In male, you will get all autosomes 44, 1 X chromosomes 45, 1 Y chromosome 46. So all varieties are available in the male sperm. So that is most suitable. So isolate the DNA from a cell. This is first step. I think I have properly gave you the information. So how to isolate the DNA from a cell you will study in biotechnology. I think if biotechnology is going on, there is a technique called as spooling. Something. Most of the techniques are there that you will study there. But anyway, let me give you, this is the first step. Isolated the DNA from the cell. Then what next? The DNA is a length of 2.2 meter. So many scientists are involved. It is not the work of one scientist to work on the whole 2.2 meter length DNA. Not that easy. So many scientists. 84 different scientists. That means it's the major contributors. Number of scientists who are involved major on frontline workers, because 84. In that, from India, eye-related genes are sequenced by Indian scientists. India also contributed to the Human Genome Project. The eye-related genes sequencing is done by the Indian scientists. So then, how each and every person will get the DNA? One cell is not sufficient. Many cells. From different cells they have to take. That means the samples should be large. And what they did, they after isolating the DNA, they by using restriction enzymes, these DNA is randomly break down into different fragments, relatively small size. The second step is break the DNA into random fragments of relatively small size. Second whole. Now what you will do with these fragments? For example, on this fragment, single scientist will not work. Many scientists will work on this fragment. A leading scientist will be there. Apart from the leading scientist, many other group of scientists will be there working only on one fragment. So if 10 scientists are there working on the fragment, that fragment must be cloned. Cloned means amplified. You need to multiply that particular, make it into more number of copies, because each and every copy should be there with each and every person in the group. So these fragments are cloned. Cloned means amplified, made into more number of copies. Where we can make that particular fragment into more number of copies, again, you have to use a specific host. The best host is bacteria. And to insert that DNA into the host, you need some vectors. Vectors are the, are nothing but the carriers which carry the desired fragment. For example, I cut down the DNA into many fragments. Fragment A, I named first fragment as fragment A. Fragment A is given to the group of scientists. Now they have to work. That fragment is first of all made into so many copies. So technically we call it as making them cloned. Amplification, which is also called as. So these fragments, which are of smaller size, are cloned in suitable host by using specialized vectors. These two are important. Host is important and vector is important. I think you got that. Now I am coming step by step, very thoroughly, giving you a good idea. You can assume with the help of explanation which I am giving you, that first you have to isolate the DNA from a cell. Most of the times the cell which is used is sperm cell. I told you why sperm cell, more DNA content. Ratio of the DNA to protein I'm talking about. I'm not saying that it contains double amount of DNA, but ratio of the protein to DNA will be more. Then after isolating the DNA, break the DNA into fragments. You can break the DNA into fragments. You have restriction enzymes to cut the DNA at a particular place and cut it in a smaller size fragments. And these fragments, distribute the fragments into the scientist group. The scientist group has maybe 30 scientists, 20 scientists. I told you, large number of scientists are required. So these fragments should be cloned, multiplied, amplified in suitable host using specialized vectors. We'll discuss about the hosts. What are the suitable hosts? The suitable hosts are bacteria and yeast. Right. Okay, this is the suitable host. Then what are vectors? The commonly used vectors to insert that into the bacteria, to insert the desired gene into the yeast are bacterial artificial chromosome and yeast artificial chromosome. Right. BAC, BAC, bacterial artificial chromosome. Then YAC is yeast artificial chromosome. I'll tell you what is meant by this. Simply this Y host, if I ask a question, Y host, the immediate answer to clone. Y host to amplify. Y host to increase more number of copies of that fragment. Bacteria, why? Because bacteria is small. Wrong. Bacteria multiplication faster. In a day, if you make the bacteria, allow a single bacteria to divide, by the end of the day, you will get millions of bacteria. Then if you insert your desired gene in the bacteria, you will get millions of the same fragment, millions of copies. That's the reason we use bacteria as a suitable host. Why yeast again? Yeast also unicellular, multiplies with a faster rate. Simple, what they do? They take a yeast cell, take out the genome of the, that means they will insert an artificial chromosome in that. In that artificial chromosome, they will have the desired fragment where they are targeting that fragment should be multiplied in more number of copies inside the yeast cell. Sometimes they will cut down the DNA at certain places of the yeast DNA and they will attach the desired gene, that is called as yeast artificial chromosome, and this is called as bacterial artificial chromosome. Right, when bacterial artificial chromosomes means they contain the desired fragment. Yeast artificial chromosome means they contain the desired fragment. Simply, if this is an yeast cell, so here this is the main genome, it will be cut and the desired gene is added here. Now when the yeast cell divide, it will be into 2 copies. So you will get the copy, this is the desired copy, it is in now 2 copies. When it becomes 4, it will be in 4 copies. It will become 5, it will be in 5 copies. This is what is called as bacterial artificial chromosome, or it is also called as yeast artificial chromosome. So any doubt regarding this, I'll stay for some time here because you might get doubts on this, so I can help you out. No problem. You can ask. Any doubt regarding this? Is clear? So, anybody from the class? What is BAC then? What is BAC?
Bacterial artificial chromosome.
What is YAC?
Yeast artificial chromosomes.
So what we do in the case of bacterial artificial chromosome, we insert the desired DNA into the yeast cell. We insert the desired DNA into the bacterial cell. Right? That is called as, so these are vectors. These are vectors. We insert this artificial chromosome and this artificial chromosome, because we are inserting the artificial chromosome in bacteria, then this is bacterial artificial chromosome. We are inserting this artificial chromosome in yeast, that's why this is yeast artificial chromosome. So these vectors are inserted into the cell. Then the cell will replicate, and these fragments also will replicate. And if you have still any doubt regarding this, you can ask. I think I have given the proper idea. Still, if you have confusion, So please, you may ask. So what is the first step? What is the first step? Hello, no problem. What is the first step? First step, yeah, first step in sequencing.
Isolate the DNA from a cell.
What is that?
Isolating the DNA from the cell.
Isolating the DNA from a cell. Okay. Second step. Second step.
Breaking the DNA into random fragments.
Breaking the DNA into random fragments. Very good. Third step.
Fragments are cloned in suitable hosts
The fragments
using
of
vectors.
glucose.
What?
Fragments are cloned in host using vectors.
Okay. Using vectors. What are the vectors?
EAC and YAC.
Yes.
VAC. And what is VAC?
Bacterial artificial.
What is YAC?
Yeast artificial. Chromosome.
Why we use bacteria and yeast?
They are small in size and replicate fast.
Small in size and they replicate faster. Can you look at the diagrammatic representation here? Is this visible? So here,
Yes,
this
sir.
is, yeah, this you can see, this is the gene of interest. Means the desired fragment. And this is a bacterial plasmid. Okay. Now this bacterial plasmid along with the gene of interest is called as bacterial artificial chromosome. This is inserted into the E. coli. E. coli. Then E. coli is grown on the media. Then it will produce many copies. So this is the ideology. Whatever the gene of interest is there, which is isolated and which is attached to a plasmid, and that is inserted into the bacteria. Any doubt regarding this? Is that clear? BAC is nothing but called as bacterial artificial chromosome. Now this bacterial artificial chromosome is allowed into the bacterial cell. Bacterial cells are competent. They can accept these bacterial artificial chromosomes. When they divide, these bacterial artificial chromosomes are also divided. This is a large piece of DNA. It can be propagated as a circular artificial chromosome in bacteria. So this particular BAC, when you insert in bacteria, this BAC, along with the main genome, it will also become replicated and pass it to each and every cell. And BAC is very smaller. The fragment is maybe 100 to 300,000 base pairs only. This BAC, total bacterial artificial chromosome. Point to be noted here, they have so many calculations. This is general idea that how many base pair containing fragment can be inserted into the bacterial artificial chromosome so that it can be again taken by the bacterial cell. Then later you have to sequence it now. You have to take out, you will get only number of copies. By this process you will get many number of copies, which is called as cloning. Then each and every copy should be isolated from the bacteria easily. That's why we use this artificial chromosomes. Right. Otherwise, if you don't, example in yeast also what they do, they usually first isolate the yeast chromosome and they'll remove unnecessary things and add this desired fragment and they make a yeast artificial chromosome and insert in any yeast cell. And you know very well, if a DNA which has a replication, origin of replication, it will replicate along with the cell. Am I right or wrong? Are you understanding what I am trying to teach you? So this is very simple. If you are still confused, I don't know whether you are not answering anything, but I am trying to help you out. But let me know whether you got the idea, concept, or let me give you some idea on this. So look at, for example, let's take yeast cell. Or let's take bacterial. This is the main genome. What I did, I prepared a plasmid where I have added the desired fragment. I allowed this plasmid to enter into the cell. So now this cell is transformed. Now this has main genome and plasmid containing the desired DNA fragment. If this cell divides and it gives rise to two new cells, so each cell will have this copy. Why? Because this plasmid is having origin of replication. So I'm not confusing you. Now this I am calling it as bacterial artificial chromosome. Right? This bacterial artificial chromosome is used as a vector. Means it is carrying my desired gene and it is taking the desired gene into the bacteria. So this bacteria is called as host. Similarly, I do with the yeast also. Yeast, what they did, I am telling you, they isolated the DNA from some of the yeasts and they trimmed that DNA. They added their fragment into the particular DNA and again inserted the DNA into the yeast cells. Then yeast cells multiply, the fragments also will multiply. This step finally will give you the number of copies, more number of copies of the desired fragment. I think I have spent a lot of time here to help you to get a very good idea. If you have any doubts, please do ask me. If you don't have any doubts, okay, you cannot answer, your mics are not working, you can raise your hand so that I can understand. I will move on. Gautaman, Geetika, Swetaram, Denilda, let me know. Raise your hand if it is completely clear. Everybody, raise your hand. Can I move on?
Sir,
Sir, if the fragment of DNA do not have any origin of replication, then how will it replicate?
Which fragment of DNA you tell me now? The fragment, desired fragment. You are talking about desired fragment, right?
Yes, sir.
Red color. See here, this fragment of DNA may not have the replication, origin of replication, but this, the one which I am using now, here, Geetika, this one, black color. This must and should have origin of replication, OR. This fragment I am adding to an artificial chromosome, this must and should have one origin of replication is sufficient. Yes, Geetika, am I right or wrong? This fragment, yes, it is not that necessary. The desired fragment have the origin of replication. Definitely it doesn't have. Most of the times it may have, it may not have. But the one you are going to attach it to it now, attached to a particular chromosome, you are making an artificial chromosome, Geetika. So in that artificial chromosome, you make sure that there are some sequences which are necessary for replication. Origin of replication is necessary. Then you will add the desired fragment to this, you will make up an artificial chromosome. Why you are calling bacterial artificial chromosome? Because you are sending it into the bacterial host. I am calling yeast artificial chromosome. Why did they call YAC? You know what they did? They have isolated the DNA from the yeast. For example, this is the DNA from the yeast. They isolated it. Okay, from some yeast cells. They have kept the origin of replication intact. They have kept some other genes also intact, and they have inserted the desired gene into this. And this yeast artificial chromosome they have inserted in the yeast. Yeast has its main bacteria, its main genome. Apart from that, one more fragment they inserted with the desired gene. This is yeast artificial chromosome. And it has the ability to replicate because it has origin of replication. This is one of the important points we have discussed. Origin of replication is very much necessary, without which, the vectors which we are using in recombinant DNA technology must and should have origin of replication. So this is a vector. If this vector is not having origin of replication, it is not possible. If it not having origin of replication, what happens? the result will be different. Let me show you that also. See, for example, this is a bacteria main genome. As you said, if this must and should have origin of replication. This is the desired fragment. What happens? I'll tell you. You will get 2 cells. Anyway, bacteria will divide into 2 cells without any doubt. Main genome, because it has origin, so it will be in 2 copies. It will be distributing, but this will be moving to only 1 cell. So now this bacteria is in 2 copies. In these 2 copies, you have only 1 copy. Every time after nth generation, you have n number of organisms. In n number of organisms, only 1 organism will be having the desired gene. So what is the use? No use. So still at the end of the day also, you have only 1 bacteria containing the fragment. The other bacteria do not have. Got it, Geetika?
Yes, sir.
Right. So is there any other doubts? So raise your hands if you are clear with this.
There is the method of transformation of the the method of transmission of the desired gene into the bacterial genome is like the plasmid transmission.
Please reframe the question. Is the
No, sir. We are inserting the desired gene into the bacteria, no, sir.
yes,
That's through plasmida, sir.
absolutely.
Yes, sir.
So we call this plasmid as why we are calling it artificial?
Not present in it naturally.
No,
That's
naturally
why.
not there. You prepared it, yeah. You prepared it. How do you prepare? You kept some genes which are necessary, like origin of replication. There are some other genes which are necessary intact, and you added the fragment. So that will become a complete artificial chromosome, no? Yes or no? It is not naturally present in any living organism. The one you have prepared right now, it is not naturally present in any organism. And here I am not going to say it as plasmid also. What I did, I prepared an artificial chromosome with the desired DNA. I inserting my desired fragment. I kept the origin of replication. I kept some other genes which are necessary, and I kept my desired fragment. I made a chromosome. What is that? Artificial chromosome. Where I am using it? I am putting it in the host. Which host? Bacteria. So what is this called as? BAC. In a similar way, what the scientists did, they have isolated already yeast DNA. From some yeast cells, they have isolated the DNA. They trimmed and they inserted their desired fragment, and again they reinserted this particular artificial chromosome into some of the yeast cells, and they multiplied along with that. This fragment also multiplied. Ultimately, we need number of copies, more number of copies. Is that clear now? Sylvia, is that? If it is clear, Ajitesh, raise your hand. If it is not clear, we'll discuss. Right, Sylvia, Abhiram.
Yes.
Okay. Everybody is done now. Right, very good. Right, let's move on because this is very important. Each and every step we have to make sure that you have a good idea. Now next step. Anyway, we got the number now. Now we can able to distribute the fragments to all the group of scientists working on. So many people working on the same fragment. That's the central idea. Then what they do? Next step. These fragments now which are cloned are sequenced. Sequenced means, now what is the meaning of sequence? Means we know what is the sequence of the nucleotides. A, T, C, C, G, C, C, T, T, A, G. That is what sequenced. So these fragments are sequenced. By using automated DNA machines. What are these automated DNA machines? See, you cannot sequence now. It is not a manual work. That this is A, then G, then C. There are certain machines which are designed by a scientist called as Frederick Sanger. This is actually once again a turning point in the field of molecular biology, where you just drop the DNA sequence. That machine will give you the complete sequence of the DNA fragment. Just drop the fragment in that machine. That machine will give you A, T, T, C, C, G, G, C, C. That is the sequence of the machine. Simple, over. So these fragments are sequenced by using automated DNA machines, okay, which are developed by Frederick Sanger. So this whole credit, goes to the Frederick Sanger. So Frederick Sanger developed DNA sequencing machines. So based on that, these fragments are sequenced. So these DNA sequencing machines, I have a point once again. Are developed on the principles of Frederick Sanger. He not only developed, DNA sequencing machines, but he also, credited for identifying the amino acid sequence in a protein. So Frederick Sanger is also credited for determining the amino acid sequence in a protein. So if you place a fraction of a protein, there are some protein sequencing machines are also there, which are developed based on the principle of Frederick Sanger. So he's also credited for determining the amino acid sequence in a protein. So the next step is sequencing. I'll come once again. Isolate, number 2, breakdown, number 3, artificial chromosome, number 4, suitable host, number 5, multiple copies. Okay, I have just divided that particular step into 3 steps. After multiple copies, each fragment sequenced by the help of DNA sequencing machines, which are developed by Frederick Sanger, also credited for determining amino acid sequence in a protein. Then after knowing the sequences, these are arranged based on overlapping regions. Once again, now you got to see once again the same word, overlapping. Sequences are arranged based on overlapping regions. Now we have to arrange all the sequences now. Okay, one by one. They are arranged based on overlapping regions. What are these overlapping regions? That also you need to know. I'll show you one diagrammatic representation. Just stay for a while, which is very important point we are discussing. Anyway, in one more class, in 2 classes, we are finishing molecular basis. We are going into strategies. Okay. Now, just show some patience here. So this overlapping regions is going to be very important. You need to know what are these overlapping regions. Check out now the overlapping regions here. AT, GA, CC, AC. Now here, this is the fragment which is starting from CC, AC, AT, CTT. Again here, this TC, TTT is overlapping with another sequence. CTC, TTT, TT, ATG. ATG is overlapping with another sequence. Then ATG, ATT, AGA. Right? Now these are from left to right. You arrange them. AT, GA, CC, AC. Right? Then ATC, TTT. Then right, TT, ATG. Right, then ATG leave it. Then you have to start from ATT, AGA. Right, this based on the overlapping sequence. What we did, we arranged the sequence in a particular order. This is completely aligned by the computer, not a manual work. Here also you can see AA, GG, AC, AA, GA. Right, again GAA, TGC, AA, GG. Again, look at here. This overlapping after GA, this is ATGC. GA is overlapping here. GA, ATGC, AAGG is overlapping. Then AC, AAGA. So come from here. So the sequence is from here to here. So based on the overlapping, no need to dive into that and brainstorm. Okay, no need to go for that. It's just simple. Whatever the fragments, whatever we have cloned, whatever we have sequenced, after sequencing, the computer has analyzed the overlapping sequences. Based on the overlapping regions, it has made a sequence. One by one, it has arranged the total genomic sequence in a particular manner. Then sequences were annotated. Sequences are arranged based on overlapping regions. Sequences were annotated and assigned to each chromosome. The next step. Then the sequences were annotated and assigned to each chromosome. This is the next step. I'll take 2 more minutes. Let me finish up. So then the homework is you have to write all the sequences in your own manner, generally what you understand by the sequence annotation, and you have to send me through WhatsApp. Okay, and the sequence of chromosome 1 is completed in May 2006. This is done. So in the next class, we will be going to DNA fingerprinting, which is the last concept of the molecular base of inheritance. And point to be noted here, annotated means assigned functions. And then where they are present, on which chromosome they are present is given. Then finally, with this step, it is done. So arrange the steps in a particular order, then you send me the flow chart of sequence annotation one by one. Right? Thank you very much. Have a nice day. We'll discuss salient features and applications of the HGP and we'll start DNA fingerprinting. With that, the whole chapter is over. Then strategies of enhancement in field production. The last 6 chapters are there, which are very small chapters only. That too also we'll have a thorough discussion on that. Thank you very much. Have a nice evening. Thank you.
Thank you, sir.
So what should we do as the homework? Please repeat.
Just flow chart of sequence annotation. First step, second step, third step, fourth step, fifth step. And highlight what are important, what is important. You give your summary. Try to now come out of the bookish kind of thing. Anyway, whatever is in the book, we studied it thoroughly. Because I am following only the book data. So I just gave you some ideas also on that. So you just develop a flow chart on whatever you have understood in the class. And give significance. You put some highlights, what you understand. This is very important step. This is challenging step. This is that. This is this. You give your summary, give your own opinion on that. That will be better. Okay.
Yes, sir. Thank you, sir.
Only Geetika has sent me that question answer. Nobody has sent. One mRNA based question I gave previously. Only one student has sent till now. Nobody has sent. So please kindly answer the things which I am giving on a daily basis. Thank you very much. Thank you. Achyut, tell me.
Sir, do we have backup classes for this lesson, sir?
Why backup classes? What happened?
Sir, I missed 2 to 3 classes in between, sir.
Definitely we'll have backup classes. No need to worry. We'll plan. So Nandini wants a backup class on Sunday. So she missed some topic. So you attend there also. I will put it in the MS team. Whenever I am taking a backup, I will mention there backup class. You regularly check. You can join, right? Okay. And whatever the classes you want, you let me know. We'll arrange once again a backup and we'll complete it. No problem at all, right?
Yes, sir. Thank you, sir.
Thank you. Gautaman, are you there? Mritika, tell me. I am fine, Maritika. Tell me how much you for 651, na? Your mother told me in the morning. Then how was? Then 8 is 11. 8 is 11, na? The mock test 10, na? 18, 18. You got it. Very good. Right. Then how the things are going on? Okay, okay. Then where actually the mistake is, it's a silly mistake or it's a kind of conceptual mistake? Can I know?
In subjects wise,
Subject wise, conceptual mistakes or silly mistakes? Conceptual mistakes are silly mistakes. It should not be done. I told already that conceptual mistakes and silly mistakes we should not do it. Okay? We should be. Now each and every exam is going to be very important. Now this is the crucial time actually. We need to play it little freely. Okay, but at the same time we need to be very conscious. When you do the question paper, you should be very conscious, which is very, very important. Huh? Very conscious. You need to be very conscious.
zoology, I felt quite difficulty regarding heart and chemical coordination chapters there.
Okay, okay.
So, when we discussed with Bharti and he took us a class, we were having a conversation.
Okay.
So, he took me the concept, since I have now. In Bharti, I do not have any problem regarding concepts. The last test I got 180.
Okay.
So, in physics, you factors like calculus, like better this test and the next test for the same chapter. So, that's the problem. But trying to solve all the questions are
Okay. Okay. Right. Then next exam you just better try not to do any silly mistakes. That is what main criteria. I was talking about only that, Meritika, because silly mistakes 15% always. Always I say 15%. If you are not doing any mistakes in water zoology, might be you are doing mistakes in physics chemistry also. So if 151, 156, it clearly says still 20 or 10, 15 marks you can add. So it might reach to 660, 670. It might get you a very good top medical college, right?
yes, yes, yes.
Please take care.
Yes,
Okay.
sir, thank you.
Very good.
Yes, sir, thank you, sir.
Welcome. Sylvia C. Yasir Ahmed. Array, you people are there or Yasir? He's just hanging on.